It's my great pleasure to introduce our next speaker, who is Folkert van Werven. So Folkert did his undergrad in the Netherlands, and then I thought it was interesting, we did a master's in applied bioscience in Japan before returning back to the Netherlands to do his PhD with Mark Timmers. His thesis work focused on the general transcription factor, TVP. He then moved to Boston to conduct his postdoctoral training with Angelica Amon, where he studied transcription interference mechanisms controlling gene expression during yeast meiosis. From there, he went to start his own lab in London, where he now leads a group at the Crick Institute. In his own lab, he has continued to study the intriguing phenomenon of transcription interference and cell state transitions in yeast, with several really nice papers on the topic, including 2 in the last week. So I'm really excited to hear the latest on his work in today's seminars, and please join me in giving a warm, if virtual, welcome to Folkert van Werven. It's over to you, Folkert.
All right, thank the organizers for giving me the chance to speak. I mean, this opportunity really arose because of a tweet from Christine to which I responded. So it's kind of a coincidence I'm speaking here. So all the data I'm presenting here are from yeast as well, but many of these processes are conserved. So I hope to present one story and maybe a short part of a second story, and both stories indeed have been published very, very recently. So transcription is highly pervasive, occurring at all kinds of places in the genome, particularly near coding genes. And this leads to a numbers of different transcriptional isoforms. And these transcription events add an important layer of complexity to regulation of gene transcription. And one function that's perhaps less appreciated is that these alternative transcription events regulate transcription of the main coding isoform in cis. So the question that my lab has been interested in trying to understand is how do these alternative transcription events control gene expression? And this has been quite a challenge. We are particularly interested in how upstream transcription influences expression from the canonical transcription start site. And actually, the Fred Winston's lab, the speaker from last week, was one of the first to demonstrate that this regulation is important for gene SIR2 in yeast. And so far, many examples have been transcribed how upstream transcription regulates gene expression. And let me give me in yeast and in mammals, and let me give you one of my favorites that we worked on in my lab together with the lab of Elchin Uno at UC Berkeley. Here's the regulation of a gene called NDC80, a key kinetochore component. Now, most of the time, NDC80 is expressed on the main transcription start site, and this transcript will encode for NDC80 protein. However, in the early stages of yeast meiosis, a long isoform is transcribed. And the transcription of this long isoform represses the expression from the main transcription start site. And it uses a mechanism using transcription-coupled chromatin changes. At the same time, the long form is under translational control. So the overall outcome that protein production is repressed. Now, the NDC80 example, I'll come back to the end of the end of the talk, hopefully. But for the main part of my talk, I will talk about how non-coding transcription regulates the decision to enter meiosis in yeast. So to introduce this topic, in budding yeast, the decision to enter meiosis is regulated by a master regulatory transcription factor called IME1. Cells that start to express IME1 will enter this program called gametogenesis phosphorylation, which consists of a round of DNA replication, 2 consecutive meiotic divisions, and the formation of 4 haploid spores. So a diploid turns into a haploid spore. Now, cells lacking IME1 will never enter meiosis. So understanding how IME1 is regulated is key to understanding how the decision is controlled. It's therefore not surprising that IME1 is actually one of the most highly regulated genes in the yeast genome. Now, you may, out of curiosity, ask yourself the question, how did I come up to study this gene? And for that, I have to be grateful to Angelica Amon, who I worked with when I was a postdoc. I would like to mention her here because she very sadly passed away last year. And many of the projects in my lab originated from my time working with Angelica. So the IME1 promoter is really an unusual large promoter for the yeast genome. It's over 2.2 kilobases in size. And this unusual large sequence is needed likely because it integrates these multiple signals here displayed upstream. First, we have nutrient control of IME1 transcription. In the presence of ample nutrients, the IME1 promoter is completely repressed. However, when in the absence of a fermentable carbon source, glucose and nitrogen sources, IME1 gets activated, and cells now can enter, diploid cells can enter meiosis. Then there's the second key regulator of the IME1 promoter, and that's the mating type signal. And this basically ensures that diploid cells and not haploid cells express IME1 and enter meiosis. And third, there are these two non-coding RNAs expressed in the IME1 promoter that we call RT1 and RT2 that basically cover the whole IME1 promoter. And they both have important function in regulating IME1. Now, I don't have time to discuss this interesting nutrient control of the IME1 promoter, but this is really important to ensure that IME1 is expressed at the right time under the right conditions. However, when haploid cells induce IME1, they will induce a lethal meiosis. And therefore, there's a mechanism exists to ensure that IME1 promoter is repressed. And this works in the following way. In cells with a single mating type, MET-A or MET-alpha, which are typically haploid cells, transcription of a long non-coding RNA that we call RT1 alters the chromatin states by bringing in histone methyltransferases like SET2 and histone deacetylases like SET3 to the IME1 promoter. These complexes repress the IME1 promoter, and as a consequence, the IME1 promoter is repressed. Now, as you can see from this scheme, there's the second non-coding RNA, which we at the time did not understand what the function of this. So the question that we had is, what is the function of this second non-coding RNA called RT2? So what I hope to convince you is that there are two functions of this transcript. First, at high levels of RT2 transcription, it represses RT1, while at low levels of RT2 transcription, it activates RT1. And I hope I convince you how that works. And this is really critical to ensure that diploid cells and not haploid cells express IME1 and enter meiosis. So Fabienne was a postdoc in my lab who drove this project, and we collaborated for modeling with Eski from UT Southwestern, and Minghao and Tsai also contributed to this project. So the first thing that we did is to determine how RT1, the RT1 and RT2 expression pattern. And as you can see from this northern blot, is that RT2 is well expressed in diploid cells, while RT1, as we expect, is properly induced in haploid cells or cells with a single mating type. Furthermore, what you can see is that, as we also expect, that in diploid cells, IME1 is properly induced. What we also see, and I will come back to that we see low levels of RT2 expression in haploid cells. Now, as you can see from the northern blot as well, there are multiple isoforms being expressed. So we first wondered whether there's something going on with weird isoforms, and we used TSS-seq, which maps the start site, and TES-seq, which maps the polyadenylation site of transcripts. And what we found is that there are multiple truncated versions of RT1 being made, which matches most part of our northern blots. So there are two important things that you need to know about RT1 and RT2. First of all, RT2 is regulated by IME1 itself, and that leads to an interesting feedback loop. And RT1 is regulated by a transcription factor called RME1. And that leads to the following. the reason that IT1 transcription is higher in haploid cells is because the ARMY1 levels are high in this cell type. In diploid cells, however, an A1 alpha repressor complex, which is expressed from these opposite mating types, represses ARMY1, and this allows for ARMY1 induction. And this ARMY1 induction allows to ARMY1 feed back to its own promoter and activate IT2 transcription. So we predicted at the time that IT2 transcription could in turn prevent ARMY1 binding, repress IT1, and promote and stimulate ARMY1 transcription. And to test this with an experiment, Fabienne expressed IT2 from a inducible promoter, the CAP1 promoter here. And as you can see, when IT2 is not induced, we see very constant levels of IT1, and ARMY1 is not induced. Now, if we induce IT2, ARMY1 is heavily induced, and IT1 levels drop. And at the same time, in these cells, the onset of meiosis is much faster. Now, I don't have time to discuss the whole story, and this was published a few years back, but we think that the presence of this feedback loop is consisting of IT2 and IT1 and ARMY1 itself is important for induction of ARMY1 expression and really increases the propensity of yeast cells to enter meiosis. Now, Fabienne thought he had figured out the primary function of IT2, but then he performed a time course experiment in haploid cells. And what he found is that prior to inducing of IT1, we see low levels of IT2 being produced. So that raised the question, is it possible that IT2 plays a regulating role in haploid cells? So what we did, the first thing we did, we make truncations in IT2 without affecting the ARMY1 binding sites, because that would mess up IT1 transcription itself. And now what we see in this truncation mutant is that first IT1 levels are much lower, ARMY1 levels are upregulated, and our ARMY1 by CHIP is reduced. So that suggested to us that IT2 is perhaps required for the recruitment of ARMY1 and activation of IT1 transcription. So we extended the analysis by inserting a transcriptional terminator in the IT2 sequence, which prematurely stops IT2 transcription. And as you can see now, a shorter form of IT1 is being generated. But what you also can see is that ARMY1 is being upregulated. So that suggested to us that perhaps IT2 transcription activates the expression of IT1. So to find additional support that IT2 transcription regulates IT1, we deleted ARMY1, because as I said in the beginning, ARMY1 regulates the transcription of IT1. So we predict a strain having a delete in ARMY1 should have lower IT1 and IT2 expression. And that's exactly what we see in this experiment. When we delete IT1, now the induction of IT2 is affected, and we cannot detect IT1, sorry. Now we can extend this reasoning, and if indeed low levels of IT2 transcription are required for induction of IT2 or IT1, then do an experiment with a heterologous promoter in an ARMY1 delete should potentially rescue that phenotype. So to do that experiment, we used the LEX-A system that has been used by published by other labs. And we integrated a LEX operator sequence upstream of IT2. And now using the system, we can induce low levels of IT2 transcription. And you can see as you have low levels of constitutive IT2, now IT1 is being expressed and induced again. So this shows that transcribing IT2 is sufficient for inducing IT1 expression. Now these results raise the question, how does this really work? Because there's been very few examples, or maybe no examples in Yeast, how upstream transcription activates gene expression. So therefore, we set out to identify potential factors that are important for this. So we did a small-scale screen and basically aimed to identify mutants where IT2 transcription is downregulated, IT1 transcription is downregulated, and IME1 expression is upregulated. And this is a summary of the data. I don't have time to show you the data, but I'm going to be focusing on this mutant or this gene called RTT109. Now, RTT109 is a histone acetyltransferase that acetylates lysine 56, the globular core of histone H3. In yeast, this modification occurs on newly synthesized histones, and it's a mark that really occurs off chromatin. It's also particularly important during DNA replication, where it can buffer transcription, and that's been shown by many other labs. And biochemically, the mark seems to be important for so-called breeding of the nucleosomes and allows access of transcription factors to the DNA. So to confirm our results from the initial screen, we did the northern blot, and as you can see is that RT1 transcription our levels are much lower by northern blot, but also by a nascent RNA sequencing approach, we see the same result. I don't have time to show you all the data that we did to confirm this, but we also used a mutant that's impaired of histone acetylation by mutating the actual residue. And we also we saw that RT1 levels are reduced and IME1 expression is increased. And it's important to note that RME1 levels, the activator of RT1, is not affected in these mutants. So next we determined whether the mark is actually present at the RT1 locus. So we performed a chip, and we found is that as you induce cells to enter, starve the cells, when RT1 is being, or IT2 is being transcribed, we saw that the mark is actually there. Now, if we use a mutant with a terminator in there, the RT2-2 mutant, the signal is reduced. So it seems that the mark is there at the right time, at the right place, and requires IT2 transcription. So as I said, this is a K56 acetylation is a mark that occurs off the chromatin. So that raises the question, how does IT2 transcription facilitate K56 acetylation? And one possibility is that IT2 transcription we thought of is that it may be due to promoting histone exchange. And there's some evidence that during transcription, old histones exchange for new ones. However, this still remains relatively poorly understood. So the question is, does IT2 promote histone exchange? And to test this directly, we used an assay that uses a differentially tagged histones. So here we have one copy of the histone tagged with a MIC tag, the other from the endogenous promoter, and one is tagged with the flag tag under control of the gal promoter. And then we basically perform an assay, and we try to measure how quickly is the inducible copy replaced at the IT2 locus. Now, we did this experiment with a strain that has no IT2 transcription or that has constitutive low levels of IT2 transcription. And hopefully you can appreciate, after gal induction, we see that the strain with low levels of IT2 transcription has a faster incorporation of the newly synthesized histone. So we think that IT2-mediated histone exchange directs K56 acetylation to the chromatin, and this facilitates potentially RME1 recruitment and activation of RT1 transcription. Now I've shown you that IT2 transcription can promote RT1 in haploid cells, while in diploid cells it represses. So how can we reconcile these seemingly contradicting functions for IT2 transcription? So we know that the levels in diploid cells are much higher. So we asked the question whether we can turn the activating function of IT2 in haploid cells into a diploid repressing function. And this again, we switch to the LEX-A, LEX operating system. And now we integrate multiple copies of the LEX-O sequences upstream of IT2. And basically increased the levels of IT2 transcription in haploid cells. And now, as you can see, is with increasing levels of IT2, IT1 levels start to decrease. At the same time, we also measure the chromatin structure at the IT2 locus by using MNase digestion of chromatin and scanning PCR primers. And as expected, when there's no IT2 transcription, we see evidence for a position of nucleosomes around the ARMY1 binding sites. Now, when we have low levels of IT2 transcription, the signal decreases. However, when we increase IT2 transcription again, we now see that the nucleosome position is positioned back again, which is indicative of a repressive chromatin state. Now, finally, we tested what's the consequence of all of this for meiosis. Now, to do this, we used a genetic trick to make diploids think that they're haploids, because otherwise we cannot actually accurately measure meiosis. The cells would die, and it's hard to measure that. And to do that, we make cells, diploid cells, look like haploid cells by making them at AA. And these cells will never enter meiosis as expected. However, if we use one of our mutants that has no IT1, IT2 transcription, these cells now are able to undergo meiosis quite efficiently. Now, if we use one of our heterologous promoters to have low levels of IT2 transcription, again, we can rescue the effect, and these cells will not enter meiosis. However, when we induce IT2 here to high levels, these cells will again do meiosis. So here's our model. So what we think is going on is that in haplocells where ARMY1 levels are relatively high, we think that low levels of IT2 promotes histone exchange to direct K56 acetylation to IT2. This in turn allows for opening of chromatin, ARMY1 recruitment, activation of RT1, and repression of the ARMY1 promoter, and these cells will not enter meiosis. In diploid cells, ARMY1 levels are lower. This allows for increasing levels of ARMY1 and increasing levels of IT2. And now this starts to make a repressive chromatin state, and ARMY1 is not being recruited, and you get a positive feedback where ARMY1 promotes its own expression. Now, we think this dual function has potentially broader implications how non-coding transcription regulates gene expression, because almost nearly 40% of coding genes in yeast express at least one non-coding transcript nearby. So to get a little bit more evidence on the level effect of RT2 transcription is a more general feature of gene regulation, we generated a data set with the aim to identify genes regulated by upstream transcription. And to make our analysis not complex like the ARMY1 locus, we focus on genes as we observed, regulated as observed by the NDCAD locus, as I mentioned earlier in the talk. And I don't have time to go to do over the details of this experiment. We basically performed a master time course of cells undergoing meiosis synchronously by controlling the expression of transcription factors, IME1, and downstream transcription factor NDTAD. Additionally, we return them to the mitotic cell cycle. So we have these three different transitions that we can measure gene expression changes since. And then we performed a TSS-seq, TES-seq to measure the polyadenylation sites and chromatin states. Now we find really hundreds of these alternative upstream transcription events. And here's an example of a gene called RAD16, which is really a remarkable changes of upstream transcription usage. Here, it's a longer isoform being generated. In transition 2, there's even longer one, while in transition 3, it switches back to a regular isoform. Now we can take all these alternative transcription events and plot them. And what we observe is that a negative correlation between upstream transcription. Basically, the more alternative upstream transcription you have, the more chances are, or the more repression you will get of the main TSS form. Now, I do not have time to discuss all the data, but if you're interested, you can read about this in our manuscript that just came out. But what we think is that in the presence of relatively low upstream transcription, two isoforms can potentially coexist and be co-expressed. However, the stronger the upstream is being made, it's more likely that switching events will start to take place. And if this upstream transcript is under translational control, this can decrease protein levels. Now, with that, I would like to thank the funders of all this work, the Francis Crick Institute, that fund the Francis Crick Institute. I would like to thank Fabienne, who just opened this laboratory in Crete. Eski, who I didn't have time to show her data, but she did the modeling part of the IMU1 regulation. And Tsai and Minghao, who helped a lot with the genomics part of this project. I'm happy to take questions.
Awesome, thank you. That was fantastic. So a reminder for questions, you can either type your questions into the Q&A box, or you can raise your hand and I'll unmute you to answer your question. While those are coming in, I have a few questions myself. So I was wondering, in your strains that have like the low or high IRT2 expression, where the low ones, it's really interesting, the low ones can actually activate transcription, where the high expression then represses transcription. Have you looked at some of the co-transcriptional methylation marks, like H3K4 or K36, over the IRT1 promoter?
Yeah, that's a good question. Yes, we looked at 36 methylation, for example, which would be an obvious one to look at. And we didn't see it there. IT2 is a pretty short transcript, so that may explain why it doesn't get there. So we screened for some of these metal transferases as well, and we didn't see an effect. So that's it's a great point. Yeah.
Then did you look at? I saw that GCM5 was in your list, like of your genetic hits. GCM5 was in. Have you looked at other acetylation sites, and if those might play a role?
Yeah, we thought about that. But we actually looked at GSIM5, and we found that GSIM5 has a massive effect on ARMY1 regulation itself. So that gave us up potential chromatin regulator. Yeah, great question, yes.
Okay, and then Jason Fan has a question. He says, hi, very cool talk. Is the histone exchange rate and K56 acetylation also up in the high IRT2 transcription state, despite seeing the strongly positioned MNase-resistant occupancy at the IRT2/IRT1 boundary?
Yeah, that's a great question. We haven't looked at that because the assays are kind of tricky. I would expect there will be some level of histone exchange there too, but yeah, it has to be figured out. I mean, this is a good question, yeah.
Okay, and then one other question was, for your K56 experiments, where you had like the RTT109 and H3K56 acetylation links, do you look at the total levels of like the bulk H3? Does that change in your RTT109 experiments? And is there like a histone deposition defect that you're seeing, as well as the change in acetylation?
Yes. Have you looked at it? I haven't checked. I mean, the cells are pretty okay. I mean, they still were able to enter meiosis. So they didn't show a massive growth phenotype. So I would expect if there's a strong depletion of histone H3, the cells would not be very happy. They were quite happy still. So, but that's a good point. If there would be, yeah, we haven't checked that.
And then I think Hanukkah has a question, so maybe she'll unmute herself.
Beautiful talk. I was just thinking more about like different like histone turnover dynamics thing, and I was wondering if you'd looked at H2AZ at all, if anything is affected there.
Yeah, we haven't looked at any other histones, how they, I mean, this is a pretty elaborate field to look at. Yes, no, this is a great question. I mean, it's something we're going to follow up on to see how that really works. And whether the whole nucleosome is being turned over or it's only histone H3, for example. Yeah, it's a good question. Yeah.
Okay.
And then, okay, so maybe one last question, which is, so like I said, I think it's really fascinating to see this example of upstream transcript rather than interfering, but actually activating. And I missed this in your second story when you kind of looked at like how broadly are there upstream transcripts. How many of those do you think might actually be activating? Like, is this how general a mechanism do you think?
Yeah, so that's a great question, and it's very tricky to look at that in this data set. So so I cannot say. We can only look at the ones that we're repressing from that data set that we generated. We tried to look because the levels are very low. So it's hard to make. The only way to really dissect that is do proper functional analysis and start to delete these upstream regions and see what happens. I think that's the only way to really dissect that properly.
Yeah. Oh, and we have, looks like we have a raised hand. All right. I will allow, I apologize, I don't know how to pronounce your name, but I'm going to let you ask your question.
Hello?
Yes.
Hi.
Hi, hello. Thanks for the great talk. I have a question, like, how do you synchronize your cells? Like, you use the alpha factor?
I didn't hear the question, sorry.
How do you synchronize your cells when you check K56 situation?
In the K56, how do I synchronize the use of cells for meiosis, or I see for the K56 acetylation? Yes. So the K56 acetylation, you have to be careful that you don't look at the replication-dependent effects. So we have to arrest them in G1 for that. And the way we do that in yeast is using a pheromone response. So we basically treat the cells with the alpha factor, which blocks the cells in G1, and then we perform the experiment. Yeah, it's a good question, yes.
Okay, so since you're using the alpha factor, does that affects the genes that you're looking at, because you're looking at the mating type?
Whether that could affect the army one. Yeah, we didn't really worry about that. We looked at this because we were looking at a wild type versus this ID2 mutant, and given that we treated them both with alpha, we didn't really worry about it. We realized that the situation is outside its natural environment, but this was the best experiment that we could come up with at the time. So we would like to take it further and study this actually in the wild type conditions during stimulation, which is not possible with the system.
Thank you.
Okay, I think we'll wrap it up there. Thank you again so much, Folkert, for the fantastic talk. That was really interesting.